Hello, good evening all. Am I audible? Hello. Okay. It's okay. You guys can unmute yourself and then answer or reply back whenever I'm asking the questions. Okay. So we can start at 6:03. Like we won't be starting the sessions right now. Uh, there are 32 people here as of now in the meeting. So let's start by introducing myself. So I'm Surabhi Chandra, a PhD student at ISC Bangalore, and as part of PMRF leadership, I will be, taking the tutorial sessions for the course Genetic Engineering. All right. So maybe you guys can very quickly unmute yourselves one by one and just let just tell your name and where are you from, which you and which college you are from, which year you are in, so that I have an idea of the audience that I have. And then today's session will be just an introductory session of what we are going to learn in this whole course. And then just a few questions, just objective type questions, representative questions, assignment questions, like how to solve them. And then slowly, as we move on to the following weeks, we will have more in-depth discussions. All right. Okay. So maybe actually there are 40 people now. So maybe very quickly just tell your name and which college you are in and what is your background, like which year you are in. Okay. So I will call out the names. Um, A is Kanwila. If you can please unmute yourself and introduce. Or if you guys are not comfortable, you can write in the chat also uh, which year you are in and what is your background. Okay. Yes, Rishita, you raised your hand. All right. Hi, Ananya. She's an MSc student. Okay. From Kerala, first year MSc. Hi, Kashvi. Okay, first year BTech Biotech from Shulini. Okay. You guys can unmute yourselves also if you wish to, or you can write in the chat also. So that just to have an idea of the audience that we have, we can proceed in that way. Today's class is anyways an introductory session. It will be, I guess, will be understood by everyone. And later on, we will be moving on to in-depth discussions from the next session. All right. Hi, Pratna. Okay, first year student, first year MSc. Okay. Hi, Madhu. BME fourth year. All right. Hi Meenakshi, first year BSc. All right. Third year. Hi Ankita. Hi Darshini. All right. Maybe we can wait till because people are still joining. We can wait till 6:10. Hi Swapnil. So and then we'll start this session. It will be a small session, maybe like an hour or so. And then from the next session onwards, we will have like session spanning 2 hours like that. All right. Hi, good evening, Sushil. Good evening, Arfa. Oh, hi, Meenakshi. No, this is actually doubt. Yeah, it can be doubt clearing session, not about this online course per se. It is basically the week zero tutorials. I think you already have, sorry, the class lectures you already have, the week one session. So just we will start with this week, what is there in the course, and just starting with a few assignment questions, we'll try to solve maybe 15 questions that they already have, and then just discuss few stuffs about cellular organelles and those things. And then we'll just end this session, and then slowly as the course next week's course starts, then again there will be, these are like tutorial sessions. I will be clearing your doubts, solving few questions like that. Okay. Okay, hi Niharika. Okay, it's 6:10. Maybe we should start now. Uh, is the PPT of first week's lecture uploaded? I think it's there, right? I think the lecture or like online lectures are online now. The series of lectures. You can uh, go through those parts. All right. Okay. All right. Maybe I should be starting now. Just one thing. Once I start sharing my presentation, the lectures, hi Prarthna, you can find the lectures in your, there's that online link, right? You can sign in, and in your Swam portal, you can access the course. And there, I think you have like week one lecture and all those things are there. So week one lectures are online. So you can go through that. Right? There only, wherever the online Swam portal, genetic engineering course, that link, you can access the course videos from there. All right. Okay, maybe I should start sharing the screen. Also, once I start sharing the screen, I won't be able to see if anyone is still coming, or I won't be able to see your chats and all. So just feel free to unmute yourself and let me know if someone is waiting in the call, or you, if you have any doubt, or if you can't hear me or anything like that. Okay. So I'll just start sharing my screen. My screen is visible, right? I will start the presentation. So basically, this course focuses on the genetic applications of biotechnology and on genetic engineering. So, what can be the different applications, biotechnology applications that we can have using genetic engineering. Like we can tweak biological systems and organisms to perform specific tasks, or change the organisms in such a way, direct the organisms in such a way so that they produce valuable substances. All right. We can also use genetic engineering to address global issues like food security, diseases, climate change, and also harnessing energy. And also we can genetically modify organisms or use molecular biology applications for healthcare improvement. We can also, have biological enzymes that can be used to enhance or upgrade the industrial processes or industrial output in large quantities. We can also take, we can also use natural microbes or uh, like bi- biological microbes or organisms to help in environmental cleanup. Basically, it expands all fields of life, whether healthcare, agriculture, and industrial applications. All right. So to start with, I guess most of you might be aware of gram staining. So basically, in this week's lecture, I will be summarizing firstly this week one's lecture, whatever were important parts, and then look into some of the questions, like sample questions, and then we will be ending the session. All right. So in gram staining, what happens is basically there are two types of bacteria. One is gram positive, one is gram negative. And gram positive means there's a particular stain called gram stain. Okay. Christian Gram, it is named after him. So gram positive are those bacteria that retains this particular stain, and gram negative are those bacteria which cannot retain this particular stain. So what basically happens is, you have this purple dye, crystal violet dye, that you apply on a smear of bacterial cells. All right. And then you apply iodine, which is a mordant. Mordant is basically something that fixes the dye. Okay. And then you wash the smear with alcohol, which leads to decolorization. Some of the bacterial cells are decolorized and some are not. And then you apply another stain, which is called counter stain of some other color, usually saffronin, which again colors the smear. So some of the bacterial cells can retain the gram stain, which is crystal violet in color, and some of them are like pinkish in color, which retains the fresh counter stain. Okay. So basically what is happening is, first we are adding the crystal violet dye, which is, so both the cell walls of gram positive as well as gram negative bacteria can affix the dye. Then the dye complex is trapped in the wall, but in the gram negative one, there is no effect of iodine, because iodine is a mordant which can basically fix the dye in the cell walls. And then upon further treatment, crystals still remain in the cell wall of gram positive bacteria. However, upon treatment with alcohol, what happens is the outer membrane in gram negative bacteria gets weakened and the dye is lost. Okay. And then the counter stain, that whatever we are applying here, in this case saffronin, is this colorless cell which got washed off due to the alcoholic wash. What happens is the counter stain is retained by the gram negative bacteria, whereas gram positive still retains the initial stain that we had applied, which is crystal violet in this case. So why is it so? Why are these two types of bacteria showing different kinds of behavior, like different um, capacities to hold the dye? Basically, this is due to the composition of the cell wall. Okay. So what happens is, in gram positive bacteria and gram negative bacteria, gram positive bacteria has a thicker layer of peptidoglycan. All right. And whereas in gram negative bacteria, the peptidoglycan layer is thinner, comparatively thinner, and it has an external surface, external layer of lipopolysaccharides. All right. So after peptidoglycan, which is actually getting washed off during alcoholic wash. That's why it's not able to retain the crystal violet dye that we are applying initially. All right. So when we talk about peptidoglycan, So what is it basically made up of? Okay, so it is actually a rigid mesh-like structure made up of polysaccharide units. Okay, repeating disaccharide units making up a polysaccharide chain. Basically, the repeating units are NAG or N-acetyl glucosamine and N-acetyl muramic acid. All right, and these two are connected by this type of glycosidic bond, which is beta 1,4 glycosidic bond. All right. And next comes this part of a cell, which is cytoplasm or cytosol. Cytoplasm is basically the area between the cell membrane here and the nucleus between the organelles. And cytosol is basically the gel-like, this aqueous um, substance in the cytoplasm, which has, salts, minerals, and organic molecules, except the organelles. All right. Then what happens here is there is the nucleus of the cell, which again are two types. Like the eukaryotes have a proper nuclear membrane, whereas prokaryotes don't have a distinction between the cytoplasm and the nucleus of the cell. All right. So in general, what does nucleus do? It stores the cell's hereditary material or the DNA, and it can coordinate various cellular activities which are really important, like protein synthesis, cell division, growth, and a host of other functions. All right. So basically, you have this nucleus, and inside you have these perforated membrane. These gaps here are nuclear pores. Okay, and there is this network thread-like genetic material uh, spread all over the nucleoplasm, which is called chromatin, which is what condenses during cell division, what we call as chromosomes. All right. So and then gets divided, and all those cellular processes take place. So also in general, you see this thread-like structures. So it can have different kinds of condensation at different points of the cell cycle. Okay. So two types basically. One is the inactive form of chromatin or called heterochromatin. Another is the euchromatin or active form of chromatin. Okay. So heterochromatin DNA is usually not very active. It is a compact form. And in interphase, this is the cell cycle phase, in this which where the chromatin are compact. And heterochromatin has long stretches of repeat sequences called satellite DNA. And also euchromatin is generally more active, and they are not very condensed or tightly packed, and these can actually be encoded, or these can actually encode for, these can be transcribed as well as transcribed to form RNA, as well as the RNA can further be translated to form proteins. Okay. Now we talked about the nucleus. Now what we saw the perforated nuclear membrane, all right, which were nuclear pores. Now, so if we look closely at a nuclear pore, it actually comprises of several entities that make up a nuclear pore complex. Okay. So basically, nuclear pore complexes are channels through which very small polar molecules, ions, macromolecules can travel in and out of the nucleus and the cytoplasm. All right. And depending on the size, molecules can travel through this particular complex, the nuclear pore complex, by either of the two ways. One is passive transport, which does not really require any energy, okay, or rather it takes place through diffusion, where there is a gradient uh, role to play, concentration gradient role to play. And another is active transport, which is energy dependent, usually against the gradient. Like from suppose a particular or salt, if some salt molecules have to be transported across, so basically ideally it should move from a concentration of high gradient to low gradient, right? However, in active transport, if the opposite has to be done, then energy is required. Okay. Next you have, this is mitochondria, which is basically 1 to 2 micrometers in length and around 0.5 to 1.1 micrometers in width, and it is a dual membrane structure where the outer membrane is continuous, whereas the inner membrane has these foldings here, which is called, these foldings are called cristae. Okay, there is this outer membrane, these are the inner membrane, and definitely this is the ATP energy currency of the ATP is the energy currency of the cell, and this is the ATP factory, right? And also it does have its own ribosome as well as its own genetic material. Okay, so the chemiosmotic theory actually explains how ATP is generated in the mitochondria. So what happens is most of the ATP synthesis in respiring cells comes from due to the electrochemical gradient across the inner membranes of the mitochondria, and there are these factors NADH and FADH2 which are formed during the oxidative breakdown of energy rich molecules like glucose. Right, you have that whole respiratory glycolytic pathway as well as Krebs cycle where it is actually these molecules are formed, right? And then ATP is synthesized basically due to the proton motive force, which is developed due to the proton gradient, and then the movement of protons or H plus ions across the membrane, mitochondrial membrane, which actually provides the energy for for ATP synthesis. Okay, so basically, see, so there are four enzyme complexes, okay, across this inner mitochondrial membrane. This is the outer mitochondrial membrane, and there is this intermembrane space. Right? Now, what happens is these four enzyme complexes are located at the inner mitochondrial membrane, which forms the electron transport system. Okay. So, what happens is we have the two molecules, NADH and FADH2. So, NADH, hydrogens from NADH and FADH2 are separated into protons and electrons, like 2H plus, 2 electrons. And these electrons then enter the electron transport system. Okay. And as these electrons are moving through these enzyme complexes, okay, from one complex to the other, the energy is used to pump protons into this intermembrane space. Okay. And what happens is at the end of the electron transport system, the low energy electrons are joined with protons and oxygen to form water. And the energy is now stored in the proton gradient that results due to an accumulation of protons between the two mitochondrial membranes. Okay. Now, what happens is there is this enzyme complex called ATP synthase. Okay, what happens is this ATP synthase is basically an enzyme, which is enzymes are basically uh, protein complexes. So the protein uses the energy of the proton gradient to make ATP. Okay, so the proton gradient, how is this produced? We saw it due to the enzyme complexes present in the inner mitochondrial membrane, and due to the proton gradient, the H plus is released in the inner mitochondrial space, like intermembrane space, and the concentration of H plus builds up in the intermembrane space, and this gradient stores the potential, which is called protomotive force or um, electromotive force, and the flow of H plus down this gradient through this rotor and stator part of the enzyme in the ATP synthase helps to generate energy, okay, from ADP molecules and inorganic phosphate. All right. Next, another organelle that usually are found in plants are plastids. So these are also found in algae, which is it has like three types. One is chloroplasts, which are used to make sugars using CO2, H2, and light under the with the help of light energy or sunshine. And chromoplasts are there, which synthesize and store pigments other than chlorophyll. And there are amyloplasts, which are non-pigmented and help in the synthesis and storage of starch granules, which are actually abundant in stems and seeds, amyloplasts. All right. So now if we look at a chloroplast structure, it is also a double layered membrane organelle having one outer membrane and one inner membrane. And again, there is an intermembrane space between them. There is the stroma, which is the watery liquid containing DNA, ribosomes, and enzymes. And there's the granum or grana, which actually looks like a stack of pancakes. Okay. And each pancake is actually these, each stack is called thylakoid. Okay. And what are the again thylakoids? Thylakoids are membrane, uh, hollow membrane covered interconnected discs arranged in stacks. And then there are lamellae, which are actually connecting each stack of thylakoid, or each grana are connected, like all the grana are connected with each other using lamellae. Okay. Now, when we talk about photosynthetic reactions, there are like light-dependent reactions and light-independent reaction. So, light-dependent reactions take place in the thylakoid membrane, right? And light-independent ones take place in the stroma, right outside the thylakoid membranes. Next, when we talk about endoplasmic reticulum and Golgi bodies, what is happening is now endoplasmic reticulum can be of two types: RER, rough endoplasmic reticulum, or smooth endoplasmic reticulum. Basically, the RER has ribosomes attached on its surface, which gives its gives them the name of RER, or having a rough appearance. Okay, and the other ones, SER, do not really have ribosomes attached, so they are termed as smooth endoplasmic reticulum. So basically, this is the RER that we are talking about because it has ribosomes, these red colored ones attached on the outside. So what is happening here is it is just outside the nucleus. Okay. So ribosomes assemble proteins from polypeptides which are entering through the endoplasmic reticulum. And then proteins move through this rough endoplasmic reticulum where they are undergoing further modifications. Then there are these vesicles which bud out of the RER, or called which are also called like transport vesicles, which pinch off or bud off from the RER, and then these transport vesicles basically fuse with Golgi apparatus, the membrane of the Golgi complex, and the proteins are released inside. Now again, the proteins further undergo various types of modifications and processing inside the Golgi complex, and the vesicles containing the final product, finally furnished protein of our usage, which has to really again go to different sites of action. So these vesicles containing the final protein, finally folded, nicely folded proteins, are again pinched off, or with these also bud off from the Golgi apparatus, and then uh, they move to the cell membrane and then released outside. The proteins, these are the peptides or proteins that you are seeing here, which are released so that they can go to their target site. Okay. Next, when we come to the plasma membrane, what actually does it consist of? What it is composed of? It is having phospholipid molecules arranged in two layers. And when we talk about a phospholipid molecule, it has a head and a tail, where the heads are basically water loving or hydrophilic, and tails are like hydrophobic, which always remains away from the water. And it is kind of arranged in this bilayer structure, okay, bilayer assemblies, where the heads interact with the fluid both outside the cells as well as towards the cytosol, and the hydrophobic tails are hidden in the middle. Okay, and there can definitely be different kinds of proteins, lipids there, and this transport channels are there. There can be integral proteins in the lipid bilayer. There can be transmembrane protein traversing through both the sides of the plasma membrane. There can be peripheral membrane proteins as well. Okay. Now, uh, Now, when we want to really work with any particular type of autoclaves, or we want to separate out the components of a cell, how do we really do that? We basically fractionate the cells. When we talk about a bacterial cell, what happens is, suppose you have a bacterial culture. What we do is we have to harvest the cells. It is suspended throughout the liquid medium, suppose. So what we do is we pellet down the cells first, okay, and then we pellet down the cells and we crush it or grind it, okay, or the scientific or the correct term for it is homogenization. And then you basically centrifuge it at different speeds for different time periods, where we can actually separate each component of the cell that we discussed like earlier. Okay, so suppose if after 10 minutes of centrifugation, what happens is you will basically have something settle down, some solid stuff settling down, and you will have a liquid layer or supernatant above the settled pellet. Okay. Now that cell pellet that is there is rich in actually nuclei and different kinds of cellular debris, and the supernatant is also rich in different kinds of cellular organelles. Now you take it slowly, only separate this particular supernatant, and then again centrifuge it for much higher speed this time and a little longer time. What happens is you will again have a pellet, okay, which are so basically the ones which are heavier, whichever organelle or whichever component is heavier will be settling down at the bottom of the tube, and you will have a supernatant above. Now this particular supernatant can again be used for centrifugations for much longer time and at very high speed. Okay, and the at this step, whatever the pellet have, this pellet is actually rich in mitochondria. Okay, and if these are plant cells, then they can have chloroplasts as well. Now, when you take the supernatant obtained at this step for further centrifugation for much longer time, which is an hour, then again what we'll have is you will have like plasma membrane settling down at the bottom of the tube, and some pieces of internal membrane structures that can be there, and then you still have the supernatant left. Now when we use this supernatant to like go at really high speeds and much longer time, like 3 hours of centrifugation, what is happening is you again get pellet rich in ribosomes. Ribosomes are protein factories. Okay, usually the pellet of ribosomes look like a glass-like pellet. Like you will have something solid settling down, but it will look like a jelly-like structure, like transparent white glass-like structure. So that is the pellet which is rich in ribosomes. All right. So now what happens is usually uh, you have to pellet down. Suppose when we talked about ribosome pelleting, okay, what happens here is usually you use the medium the, through which the each component has to separate usually contains some kind of density difference across the length of the tube. So what happens is suppose sucrose is a sugar. Okay. Now different concentration of sucrose is layered on top of the other. Suppose if one is making 10% sucrose, 20% sucrose, 30% sucrose. Suppose as an example. So 30% sucrose, suppose if this tube is having a volume of total 3 ml. Now 30% sucrose 1 ml, then again 20% sucrose is layered on top of 30%. Definitely the 20% sucrose solution is having lower density as compared to the 30% sucrose solution, right? So it will stay on top of this 30% sucrose, and then the 10% being the most least dense will be staying on top of all these density gradients. Now what happens is when you want to separate, suppose your cell mixture, your homogenized lysate, you want to separate the components of the cells. What you do is you prepare this particular gradient, okay, and then you put your cell suspension there. So this blue color thing is there, resuspended on top of this density gradient. Okay. Now what happens here is, now suppose uh, uh, what we saw earlier is like nuclei separating first. So the whole nucleus, that is more um, nuclei separating first, or suppose the debris of the cells are separating first, ideally, right? So what is happening is the ones which are heavier, in general, all the components of the cell will divide according to the region, like matching the regions with matching densities. Okay, the ones which are lighter will definitely can't penetrate the higher density region. Okay, so it will stay on top where its density of the particle matches the density of the surround. Okay, so the slow sedimenting particles will stay up, fast sedimenting particles will stay down. So what happens is this is how the separation will take place. And then what you do is you basically this is uh, like this is one way of fractionating where you are taking the most dense ones will be settling at the bottom first. So you are taking the most dense ones. You basically have a perforated tube where you can take it, dropwise each layer of the whole medium. Okay. Or one can also do is okay. What one can also do is you can also take the least dense ones first. You can pipe. There are pipettes that you can take the solution out from the top also. Okay. This is called top-down fractionation, and this is bottom-up fractionation. Picture that is being shown here. Okay. So basically, the first fractions are the most dense ones, and the density decreases as you go further. But when you take it from the top, it is the opposite way. You have the least dense uh, fractions in the beginning and the most dense fractions at the end. Okay. So this is basically the same thing that we saw earlier. Okay. Okay. Now just this is now let's go to this is what the whole week's lecture covered. The lecture videos covered. So we'll look at answers are already there in front of you. We'll just want go through these. There are only 15 questions. Okay. So what is happening? So the prokaryotic ribosomes. Now, when we talk about the protein factories, so prokaryotic ribosomes are there and eukaryotic ribosomes are there. So prokaryotic ribosomes are and ribosomes are basically represented as prokaryotic ones have two subunits, eukaryotic ones also have two subunits, but the subunits differ. In case of prokaryotes, it's 30S and 50S. 30S is the smaller subunit and 50S is the larger subunit. And in eukaryotes, what you have is 40S, the smaller ribosomal subunit, and 60S, the larger ribosomal subunit. So when I'm talking about S here, S is basically the Swedberg unit. Okay, it is not regarding also when the whole machinery when the two subunits join, it forms the whole ribosomal complex. So in prokaryote, when the 30S and 50S joins, it doesn't really form 80S ribosome. It forms 70S ribosome. And in eukaryote, when the 60S and 40S joins, it forms 80S ribosomes, not really 100S ribosome. So basically, it's not an additive value. Swedberg is basically the time taken for these entities to centrifuge and sediment down at the bottom. Like we saw earlier, different components of the cells settling down after a certain time. This is what the Swedberg unit represents. And also the S here is ideally capital. Okay. So the prokaryotic ribosomes, like we discussed, is made up of 70S um, unit. Okay. Is of 70S, which is, if we divide it further, is actually made up of 50S larger subunit and 30S smaller subunit. Okay. Now, when we talk about which of the following is true for prokaryotes, presence of introns, membrane-bound organelles are present, possess single origin of replication, and transcription occurs in the nucleus. So in prokaryotes, as we discussed, like there is no division between the nucleus, like nuclear membrane is there in case of eukaryotes. You don't have that in prokaryotes. Okay. So what happens in case of eukaryotes is like you have the mRNA synthesis or transcription well differentiated, well segmented into from the protein translation. The mRNA comes out and then the ribosomes translate the mRNA to form proteins. But in prokaryotes, since there is no distinction, there is no compartmentalization as such, what happens is that uh, the term is called coupling. Transcription translation coupling happens that once the mRNA is synthesized, the RNA polymerase is synthesizing the mRNA from the DNA. Okay. And the mRNA that is coming out of the polymerase, suppose this is the mRNA that is coming out of the polymerase, this mRNA is simultaneously attached by ribosomes to form proteins. Like one mRNA is simultaneously getting, one is transcribed, and as it is getting out, that mRNA is attached by ribosomes, and then those ribosomes in turn read the mRNA and form corresponding, like join the corresponding amino acids and form the polypeptide. Okay. And also when we talk about introns are basically interfering genes. Okay. Prokaryotes do not usually have introns. Eukaryotic genes have introns, which, if you know the term splicing, which has to be, these introns have to be removed and all the coding regions have to be join together to form the to form the coding mRNA. And then this mRNA is read by ribosomes to form proteins in eukaryotes. Okay, but prokaryotes have polycistronic genes without any introns as such. Okay, and of course, in prokaryotes, we do not have membrane-bound organelles. So we saw that, okay, nuclear membrane is there, plastids are there, mitochondria are there. All those are like membrane-bound organelles. Okay, but these do not have membrane-bound organelles present, and transcription occurs in the nucleus. This is true for eukaryotes, not prokaryotes. So the correct answer is it actually possesses a single origin of replication, and from where DNA sisters are made. I mean, DNA is replicated. Next, antibiotic resistance in bacteria is conferred by, so plasmids, answer is plasmid. Plasmids are basically circular genetic material, circular DNA present inside bacteria, which actually has certain regions which encodes for antibiotic resistance, okay, which helps the bacteria to thrive in these conditions. And then, which of the following organelle is found only in plant cells? Glyoxysomes, vacuole, mitochondria, RER. So these are in general found, these three BCD are found in general in other types of eukaryotic cells as well. But glyoxysomes are found specifically in plant cells. Now we discussed about euchromatin and heterochromatin. The euchromatin, which is present in the nucleus, is actually transcriptionally active, and the heterochromatin that we talked about is not inactive. It is very densely coiled. So the all the machinery, the enzymatic complex that needs to reach the genetic material to read the genetic sequence that is there, those can't really reach there. So that's why it's transcriptionally inactive. Okay, and it can take up the stain very nicely, which is why it is, those are the dark regions that we see under the microscope, and it has a densely condensed genetic material. Okay. Next, we also read about the ATP synthase enzyme complex. The ATP synthase is embedded in there are two membranes of mitochondria. ATP synthase is basically embedded in the inner membrane of the mitochondria. But out of these, cristae is basically the foldings of the inner membrane. So the answer will be cristae. And intermembrane space actually has the H plus ions pumped inside. Okay, which is then used, the gradient difference is used to generate ATP. Okay, so dash is released by mitochondria to initiate apoptosis. Okay, so during apoptosis is basically cell death. Okay, and out of this, mitochondria ATP synthase no, ATP no, protons no. The answer will be cytochrome C. Next, we also read about the light reaction and dark reactions of photosynthesis, which occurs, which region of the chloroplast is the site for light reaction and dark reactions. One is this light reaction is the thylakoids, and stroma is the light independent reactions. Okay, stroma is the site for light independent reaction. Okay. Which of the following organelle is the major site of calcium sequestration in the cell? This is also there in this week's lecture video. So the endoplasmic reticulum is the answer, where it basically can store a lot of calcium, and upon need, what happens is we saw that it has the vesicular stuffs. It can release out upon need. When the signal comes, it can actually release out calcium ions. Okay. Also, we know about muscle contraction and relaxation, right? So muscles also have sarcoplasmic reticulum, right? So that can also release calcium upon requirement when there is a need. Okay. Okay. Degradation of pathogenic bacteria inside the cells occurs in the secretory vesicle, phagolysosome, RER, SER. Answer is phagolysosome. So lysosomes are basically suicidal bags, okay, which have the lumen of the, basically these are vesicles, and the lumen of the lysosome are really acidic, having a very low pH, where these pathogenic bacteria are trapped inside those lysosomes, and it can help in killing the bacteria inside that has been trapped. So that's why it's called it the lysosomes that actually eat up those bacterial cells. That is why phagocytosis. Okay, and then forming these lysosomal bodies which can digest the bacterial cells. Okay, so the answer is phagolysosome, or answer B. Next, when we talk about antibody secretion, antibody is secreted from a B cell. So, which is the pathway the protein is followed? Antibody is basically a protein. Okay. So, we looked at the pathway in the previous slide. So, the answer will be very first, there's outside the nucleus only, there's RER, then it goes into Golgi for further modification, and then the protein, whichever protein. In this case, we are talking about antibody, but antibody is basically a protein. So then it is secreted out inside a secretory vesicle. Okay, like from the Golgi apparatus, the secretory vesicle reaches the outer membrane, and the peptides or proteins are released. Okay. So the answer will be ARIA followed by Golgi apparatus followed by secretory vesicle. Out of these options. Now we just discussed about phagolysosome. So the lumen, the cavity of the lysosome has a pH of, out of these, we talked about the lumen having an acidic pH, very low pH. So, the lumen of lysosome has a pH of 4 to 5. Now, isolation of proteins from the periplasmic space of bacteria is done with. So, what happens is, again, all these questions that we are going through, each of this step will be dealt in detail in the later chapters. Okay, this is just a brief brief trailer of what we are going to read in the subsequent weeks. Okay, so uh, in bacteria, what happens is periplasmic space is the space between the cell wall and the plasma membrane. Okay, so in that area, what happens is we, what we saw earlier is we were centrifuging to separate the different components of the cells, basically the different organelles and all those stuffs. But what about the proteins which are present in the space outside the plasma membrane but inside the cell wall? All right. So what happens is these are usually used to take out proteins or nucleic acid or ribosomes from the intracellular space. Okay. But outside the space, what happens is we have to basically take the proteins out from the periplasmic space, so the cell wall should burst. So what happens is you give osmotic shock. What happens in osmosis is basically the liquid flows from a higher concentration gradient to a region of lower concentration gradient. Okay. So what happens is when the liquid gushes in, the cell wall actually bursts open. That can actually release the proteins that were inside the cell wall but outside the plasma membrane. Okay. So the answer will be B, osmotic shock. Next, you have pathogenic bacterial cells are killed by reactive oxygen species, which are released from the dash of immune cells. So, these reactive oxygen species, the organelle which is the source of reactive oxygen species in the cells, are the mitochondria. Okay, so the answer is B. So these are actually endoplasmic reticulum, Golgi bodies. All these are sources of protein like sites of protein modification, protein folding, and all those stuffs. Okay, but mitochondria is the site where reactive oxygen species um, are released, because you have all this electron transport chain going on, all these redox radicals are formed due to the different processes going on inside the mitochondria. Now, during apoptosis or cell death, the genetic material of the cell is degraded by, now all these are enzymes. Okay, names of enzymes usually ends with ASE, something ASE. Now, when we talk about the genetic material of the cell, which is ideally DNA, fine. And what is DNA? DNA is a nucleic acid. So, any kind of enzyme that is degrading something, we usually add an is in the end. Okay, like any kind of action is happening. So the term protease, this is an enzyme which actually degrades or acts upon proteins or peptides. Okay. Then there's ligase. Ligase is actually, it acts on, it actually helps in joining two DNA ends. Okay. So it is not really degrading the DNA, it helps in joining two DNA ends of nucleic acid. Okay. Lipase are enzymes which acts on lipid molecules. Okay. So basically the genetic material is DNA. The enzyme which acts specifically on DNA nucleic acids are called DNAs. Okay. Uh, just a small note is that when you write DNAs, the enzyme that acts on DNA nucleic acid, you write it as this, like capital D, capital N, and then is, which is basically representing an enzyme. Now, when you talk about, usually we don't, but still, when you write like proper DNA, capital D, capital N, capital A, then you add like small s, it basically represents plural term. But this is actually um, an enzyme that acts on DNA, nucleic acid. Okay, so this is it for today's class. Okay, so I will stop sharing the screen. And so this was a very short class today. From next week onwards, so each of the steps that you saw, like the differential centrifugation, how are we purifying proteins, we saw some terms. So all those, and what are the different steps or molecular biology techniques that we apply, what is the basis, what is the principle of each technique, how are we really doing genetic engineering, how are we genetically modifying a particular organism or a bacterial cell or eukaryotic cell, how are we really doing that? All those we will be looking at in the subsequent weeks. Okay, this was just an introductory class, and this is how the assignment sessions will be, where you basically have, we will be looking at the previous week's questions and trying to solve and basically go into the conceptual part of the question and then try to look at the answers of it. Okay. Okay then. See you all in the next week on next Saturday from 6 o'clock. Thank you for joining. Thank you so much. Also, I think someone in the chat is asking how to get the lecture notes. I looked at the site. So the lecture note will be there. I think week one lecture notes, you will have all the session notes there, the slides there, and also today's slides also I will be uploading, and the link will be shared in the spreadsheet that you have from where you got the Google Meet link also. So there will be all the this recording, today's sessions recording and everything, and as well as the slides will be there. So all the link and everything will be shared, as well as this whole recording will be uploaded in the YouTube. So that YouTube link will also be provided to all of you. Okay. So don't worry, like everything will be shared with all of you. Okay. Okay. See you then. Thank you. Thank you so much. Thank you. So I will end the meeting for all of you.